Thank you, Anne. It's a pleasure to be here to kick off this BioProcess International Theater event. And we're looking forward to a robust discussion on how we can speed development, lower costs, and enhance quality at the same time, what we're calling the Trifecta. And the reason we came up with this phrase was that the conventional wisdom in the industry is we have our triangle of quality, speed, and cost. And you can choose 2 out of 3. So you want a high-quality product and you want it fast, well, it's gonna cost you. Or you want a high-quality product and you want it to be inexpensive, that's going to take some time. But increasingly, we as an industry are being asked to do all 3. And as the folks who have to actually make this happen, we want to know what's driving this trend and how can we deliver on this? What type of technologies and approaches can we use to actually do things quickly in a low-cost way and still achieve the quality objectives? So I thought as the moderator, I'll just give some introductory remarks on the driving forces and also some of the challenges, and then we'll introduce the panel who will go into some of the innovative and exciting approaches that we as an industry are looking at for delivering on the Trifecta. As most of you know, we're all being asked to compress our timelines continuously, and what's driving that? Well, we're seeing a number of things in this field that are really forcing us to look at doing things faster and faster. First of all, there's competition in a number of hot discovery areas. Certainly the PCSK9 antibodies are one good example. The immune oncology field, there are enormous numbers of products competing to be first to the clinic and first to the market. And along with that has come new regulatory pathways. Breakthrough therapy designations have allowed us to compress our clinical timelines, which is great, but we also have to compress our CMC timelines along the way. Although very different products, biosimilars face the same challenge of needing to operate on very compressed CMC timelines. So speed is becoming more and more important. What's driving cost? Well, obviously, as an industry, we've grown to now the point where pharmaceutical sales are forecast to reach $1 trillion right about now. In the U.S., healthcare spending is about 20% of GDP. And there's increased pressure on pricing from the cost of healthcare overall, as well as the emergence of biosimilars competition as this becomes a major force in our industry, which is certainly becoming. I show here the number of biosimilar products in development for some of the major antibodies that are coming off patent in the coming years. And there's an awful lot of competition in this area that is certainly going to drive prices down. Couple that with the fact that we now have technological capability to bring our costs down, and we're being asked to deliver products at lower costs as well. And finally, why quality? Well, quality is always important. We're making products that are being injected into humans. This is a critical aspect of our work that we can't skimp on. And along those lines, we're also in this big paradigm shift. For those of us who started in the industry 30 years ago, we started with the mantra, process defines product. Whatever the process made is our product, and we couldn't characterize it well enough to really change that easily. We're moving to defining our product ahead of time, the quality by design mindset. And as we make that transition, the emphasis on quality, understanding your critical quality attributes and how we can control our processes to deliver that becomes more and more important. So the reality is we need to be able to deliver on all 3 of these areas, quality, speed, and cost. And as an industry, we can do a lot with fine-tuning. We can implement operational excellence initiatives, we can tweak our platforms, we can make improvements in like-for-like areas, better membranes, better resins. But really to make leaps forward, we sometimes have to take the jump to adopt novel technologies, truly novel approaches. And some of the examples that you'll hear about today include continuous processing, creative management of supply chain so we can leverage expertise across the industry and not just within our organization, and real-time quality, process analytical technologies, and even potentially real-time release. But implementation is always a challenge, and that's been true in our industry, and it will probably continue to be true. Why is this? We as an industry have been able to adopt new technologies. Parrish will talk about single-use technologies, and that's a good example of one that's been adopted well over the last decade. But we're very conservative, and there are good reasons for that. There are significant costs and risks for implementing new technologies. Nobody wants to go first. The penalties for delaying a program because you're taking a creative approach are severe, so few people want to take that risk. And there's really no good time anymore to implement a new technology. We want to move fast to the clinic and fast to market. So we're challenged to find good times to do this. And finally, as an industry, we invest relatively small amounts in new technology development, and that may be an area we want to consider collectively changing. Is if we're really going to make these leaps, we have to invest more in new technologies as an industry. So how can we improve implementation in a way that minimizes the risks and avoids delays on critical development programs? Well, I'm not going to answer that question. That's going to be our panel. So I'm going to introduce the panel now, and we'll hear 4 excellent talks on how we might do this, starting with Joanne Beck, Senior Vice President of Process Development at Shire. Parrish Gallagher is CTO Upstream of Accelerex GE Healthcare. Lynn Frick, Director of Sales, Continuous Bioprocessing for the Americas at Paul. And finally, Mark Brower, Senior Research Chemical Engineer at Merck. These panelists will give approximately 10 to 15-minute presentations, and then we hope to have a good panel discussion following the talks, where I'd like to encourage everybody in the audience to ask questions so we can have a good discussion that includes all of you as well. With that, I'd like to introduce Joanne. Joanne has a PhD in biochemistry. She brings a really strong scientific as well as practical aspect to process development. Many years of experience at Genentech, Amgen, Abbott, and now Shire, where she's Senior Vice President of Process Development. So please join me in welcoming Joanne.
Hello, everyone. Thank you, Tom, for the introduction. So my talk is going to focus more on general approaches of how to get a pretty diverse pipeline, products in a pretty diverse pipeline as fast as possible to the clinic and hopefully eventually to the market. So because our focus is in rare diseases mostly, but also specialty conditions, we have a very, very diverse pipeline in terms of technology platforms. We are looking at early development programs in gene therapy, also in MRT, which stands for mRNA replacement therapy. So the drug substance is actually mRNA that is synthesized through enzymatic process. Traditional proteins, which is the core of Shire, enzymes, and non-antibody proteins. We have a number of antibody products in our pipeline, and also we have a plethora of small molecules. So our R&D group is using internal innovation to develop programs, to discover and develop programs. But we also have a very broad spectrum of partnerships in each of these areas that are shown underneath. These are examples. For example, we have a partnership with Sangamo with gene editing, gene therapy products, Ethris and the Cystic Fibrosis Foundation for our mRNA replacement therapy products, et cetera, et cetera. And so we took in pharmaceutical development a very similar approach, where we are also supplementing our internal capability for developing processes and analytical methods for all these different technology platforms with external partnerships. So anyway, so the development landscape at Shire, looks like this, and I think it's pretty typical, right? We have the BioProcess We use mammalian cell culture, both CHO cells as well as human-derived cell line that most of our enzyme replacement therapies are manufactured. We use single-use bioreactors and tanks, and the harvest is continuous through continuous centrifugation. We also have products that use bacterial fermentation. We do not have any internal capacity for bacterially derived proteins, so that is partnered out. We have a sort of a hybrid model for these partnered out programs, the bacterial, where we do some of the analytical methods internally and we transfer them to our CMO. We also have viral vector production, again, that is done externally, and all the purification is non-platformable because everything is different. There's not like a monoclonal antibody where everything's normalized through protein A capture step. Formulation, drug and combination product development, the landscape there is equally complex. Our formulations are not really platformable either, and that's specifically because we have pretty limited protein engineering options where you can take an antibody and you can engineer it more solubility or less solubility if you so desire. And in this case, we're kind of stuck with the human version of the protein. The excipients, because these proteins can be very insoluble, hydrophobic, we have to use some non-traditional excipients, so there's quite a bit of work that our group does internally in this case to develop formulations for these difficult to formulate products. Gene therapy is a new modality where we're trying to figure out what the optimal formulation is there, and then we are also using an intrathecal delivery platform that we developed in collaboration with a company that makes intrathecal devices. In terms of analytics, all our analytics have to support all these multiple different platforms, and we're very focused, especially early in development, not high throughput, but also high resolution. So there's quite a bit of work that's been done by the group, the analytics group, to develop high throughput, high resolution methods. Of course, with all of this data coming from different platforms, internal, external, manufacturing, pilot plants, etc., quite a bit of work is done to acquire the data and then aggregate it and analyze it. This is actually interface with IT is a huge project. So just a little bit more on the different platforms and the approaches that we have taken, because depending on the platform, you change the approach. If our optimal business model at Shire is to outsource as much as possible so that we do not build and own and have to run internal capacity. So the far right, you see the small molecule model. Everything is outsourced. Development, analytical method development. We don't have any labs we do not own any manufacturing capacity. So we would like to apply that same approach to gene therapy and mRNA replacement therapy. These are new platforms, and we do not want to invest in hiring a ton of people, building a facility, because these are not proven really platforms until we have proof of concept. We've gone through a successful phase 2-3, and then we can consider commercializing and building capacity. However, the problem is that the market out there is not really mature, so there isn't really a lot of very expert contract manufacturers or even contract development organizations that we can partner with to develop and manufacture gene therapy products and mRNA replacement therapy products. So there we have to really supplement the external capability with internal know-how and expertise. So require very, very intense oversight in order to develop. So in these cases, we really truly leverage biologics expertise, and I speak very broadly. For example, vaccine, we have people that have worked in vaccines at Merck that are now working for us, and they're leveraging that experience that they had with vaccines to do the gene therapy platforms. And of course, with the proteins, that's our internal platform. We have a lot of internal expertise in that, so we keep it inside. For monoclonal antibodies, the antibodies that we have in the pipeline, we plan to completely outsource everything and basically have a project manager and some key CMC leads, but that's it. So because the monoclonal antibody, it's almost like small molecules at this point. There's a lot of expertise available externally. In terms of how to move faster and with higher quality, I put the example of our internal manufacturing where we used our bioreactor platform. We started with stainless steel, or in some cases roller bottles, and then we had to expand capacity and get really fast to market. So we adopted the single-use technologies. It allowed us to build and bring, IQ or QPQ, get the program in, the product in, and validate, etc. Very, very fast. So that was a solution. The expandable shell allows us to add capacity as needed over time. So that was a way to actually get. So this is what we're looking at when we partner. Is our partner capable of expanding quickly? If we need capacity, can they bring capacity online very quickly? How flexible are they? So in terms of dealing with numerous platforms, I don't want to go through every single bullet point in this slide, but basically, up front, we have to decide whether we're going to do it internally or externally. In general, we try not to do things internally until a particular platform or product is de-risked. Many cases, we use a hybrid model when we have internal expertise, for example, a high resolution analytics. We tend to do it internally and then do the tech transfer. In both cases, you need a lot of internal expertise. The idea that you can just outsource a biologic is not really realistic. You're going to have and just let it go. You have to have continuous oversight. In terms of maintaining not just speed and cost, but quality, implementing the principles of quality by design up front, whether a program is internal or external, is also extremely important. And we make sure that is very clear, and all the information and data and all the experimentation and the development, whether it's internal or external, feeds into our internal quality by design based templates. I'm not saying that we're going to do a quality by design filing. I'm just saying that the whole concept around process and product understanding and process control is implemented up front, regardless of whether the product is made internally or externally. So that ends my talk.
Thank you, Joanne. We'll hold the questions till the end to the discussion period. I think that was a great example of how a company with a really diverse pipeline is using strategic supply chain management to optimize the development approach. And our next speaker is Parrish Gallagher. I'm not sure Parrish needs an introduction, but I'm going to give him one anyway. Parrish has a master's in chemical engineering from MIT and started his career at Biogen, moved on to Alpha Beta, and then to Millennium, where I actually worked with Parrish in one of my earliest consulting assignments on a technology called Bugs on Wheels at the time, which was the nascence of what became Accelerex. So Parrish was Chief Technology Officer and founder, and is now Chief Technology Officer at Upstream for Accelerex GE Healthcare. Please welcome Parrish.
Thank you, Tom, for the introduction, and it's an honor to be a member of such a distinguished panel of industry experts. So I'm going to carry on from the previous two presentations and talk about the trifecta opportunity in innovation in biomanufacturing, in which benefits in cost, quality, and speed, all three can be achieved. So, and what we're seeing is that this is really an exciting time of innovation, and I like to call it sort of the new era of innovation in cost, quality, and speed, because the stage has really been set for the opportunity in the fact that we have higher titers today, we have single-use technology, and in a lot of our new facilities, the scales are not so large. So the implications of what we do and the cost and speed to install them are shorter and lower. So on the left, we have the pressures on us as biomanufacturers as a result of all of the market trends that Tom reviewed. And so we need to get higher productivity and tighter yield out of our processes, but that's not enough. We see folks pursuing better plant utilization, more flexible and agile facilities, reducing variability in the form of reducing failures, reducing waste, running at higher efficiency in their operations, operating in niche markets and at smaller scales, which of course works against economies of scale, working against cost. And at the same time, another pressure on biomanufacturers is the increasing expectation of product quality assurance in manufacturing and a better understanding of product quality attributes by not only us, but also by the regulators. So there's no break on quality in this new world. So what's going on? How are people responding to these pressures in our industry? And what we're seeing again is an avalanche of innovation activity where cost, quality, and speed are all being attacked through very creative ideas and approaches. And I've really been excited and impressed by many of these. In many cases, there are multi-pronged approaches where there's a synergistic impact, and that's what we're looking for. We're looking for the synergy of cost, quality, and speed benefits, all three with our innovations. And we don't just see these in small companies that are more innovative, we think. We see innovation programs in large companies around the world. And again, enabled by higher titers and single-use technology. So what that means is, in order to be in the game, you pretty much need to be in single-use, and you pretty much need to be at higher titer in order to be competitive in this new world. So assuming that you've made that grade in your organization, we're going to talk about some of the other technologies that are represented here as to what in the rest of the process that people are pursuing and looking for that trifecta. A lot of these new technologies are obviously happening in the development arenas and in laboratories and pilot plants. So the commercial viability obviously needs to be verified over time, and that of course will happen. And the good thing is that a number of regulatory agencies get it. They get the need for innovation. They're not driven by cost and speed, but they're driven by quality. So innovations that target quality assurance, maintaining product quality during manufacturing, really ring up with the regulators. Okay, so let's take a look at just a quick review of single-use technology. So again, to be in the game, you have to be in an agile, flexible state with your operations. Single-use can get you there for many applications, not all applications, but many applications. And today we have a whole list of tools to use that are single-use, and fully integrated flex facilities are now the opportunity and are being built around the world in many different countries and by many different companies for many different types of products. So that's the reality today. Well, what's the report card on single-use technology? You know, what are users really saying about it? What's the bottom line about the impact of single-use on their costs and their speed? Well, these are the companies that I've been watching, and they've been reporting over the last 5 years, and I've summarized their data in this chart. So if you look across the top, again, these are customer reports that have come in over the last 5 years about single-use savings versus a traditional approach. And as you can see across the top, the cost of consumables, facility capital, footprint, labor, time to build, turnover time, water use, energy use, carbon footprint, and increased annual capacity. And the last column is operating costs of goods. So the data from each of these companies, the companies are listed on the left, and their data is filled in the chart, as you can see. And the bottom line is the bottom line across there. So at the average, at the bottom of the chart, we can see that our consumables are increased by threefold or so. Our facility capital costs are cut by almost 50%. Footprint by 23%, labor 33%, time to build a third, turnover time sped way up, water consumption and energy consumptions are reduced, and the carbon footprint is also reported to be reduced in a number of companies, with increased capacity reported by some. All rolling up into an operating cost of goods reduction of about 27% on average, which includes the cost of the consumables. So yes, the cost of the consumables go up by threefold, but those additional costs are overwhelmed by all the other savings that you see across the bottom in all those other categories. So that's the report card so far from users on single-use technology. We've also enjoyed the benefits of high titer. So CHO cells are generating gram per liter concentrations, and as you can see, that brings you way down on the cost curve into the, on the order of $100 a gram at a significant scale if you're in the 3 to 5 gram per liter range. And that's where most folks need to be, especially in the biosimilar category, to be cost competitive today. So single use and high titer, great advances in cost and speed. And single use also gives us the quality advantage in eliminating cross-contamination in our facilities. So that's another thing that the regulatory agencies really like, eliminating cross-contamination possibility. But we don't think, we see that in a lot of companies, high titer and single-use technologies are not enough. They're going beyond those two advances. So what are they doing? Even though there are facilities around the world using single-use, running at high titers, they're still examining new innovations to increase utilization and efficiency. So the question will be, which of these tools would you use in your organization? And there's no easy answer, and there's no one answer, right? It really depends on your manufacturing strategy. So the manufacturing strategy is the first thing you ought to lay out, and that'll help you define which biomanufacturing innovations to further pursue and which of those tools should I actually deploy and use in my process. So you need to look at the dose, size of market, the kilograms you need, the titer you're at, the COGS target that you're shooting for, the stability of your product, the scale of the operation, how much experience your team has, how much automation experience your team has. Are you going to insource or outsource? Location of your facility, which territory, what are the regulatory and skilled labor pools there and environments there, and are you going to expand later? And by the way, how much money do you really have to spend, right? So the strategy lays out really where you're going to go and what things you ought to try and how far you want to reach in terms of your innovations in your process. So what are some of those examples? So process intensification, of course, a lot of people are examining process intensification in the upstream space. It includes continuous operations or continuous with cell recycle operation into the bioreactor, bringing the cells back into the bioreactor. There are a number of tools here shown on the screen for recycling cells back to the reactor. And in these cases, you either have to do filtration before column chromatography, or in the case of a filtration system for recycling cells, you get a clarified permeate. That permeate can go right on your column, so you don't need to further clarify the cells out of the process. So that's a tool. These are tools available to you for perfusion and intensification in the upstream. This shows some data simply from one of those examples, where in this experiment reported by Crucell, they got up to 60 to 70 million cells per mil in a perfused bioreactor, single-use bioreactor system. How about going downstream? Well, a lot of folks are looking at the tools associated with connecting or integrating and combining steps into straight-through processing. This is an example reported by the fellows at the folks at J&J Centocor, in which they eliminate the intermediate hold steps such that they connect all the processes together so that they run semi-continuously in a connected fashion to shrink the batch cycle, the overall cycle time, and eliminate holding tanks. And in some cases, they use inline buffer dilution, so they simply make concentrates of buffers and dilute them online, so they can shrink the buffer storage tanks as well. So here's another innovation that's being evaluated in a number of laboratories and pilot plants around the world. Another innovation is continuous chromatography, and we're going to hear a lot about that from our dear friend Lynn, an expert in this field. And there are a number of systems available to you for continuous chromatography, in which you can use multiple columns, and the benefits here are reduced buffer consumption and resin consumption. And in addition, some people are coupling these systems with inline dilution to, again, use buffer concentrates. Another innovation, or innovations that people are using more and more widely, are plug-and-play closed processing systems, such as pre-packed columns, which are available from a number of folks, and also cartridge systems available from Natrix, Paul, Sartorius, and others. And these afford the ability to quickly set up a process, connect it, have everything closed, so you can do closed processing, which can reduce the atmospheres in your facilities or environmental controls requirements, and then quickly break down for disassembly and turnaround time. So very convenient plug-and-play processing with closed cartridge systems. The innovations we see don't just have to do with the process. What we're finding out is that a lot of folks are saying the need to change the facility as well. So another way to increase utilization and efficiency is to look at how your facility is designed. So on the left of it has a lot of clean rooms, a lot of gowning rooms, a lot of corridors to get things in and out of, especially if it's single use and you have to carry them all in and you have to carry them all out when you're done. The more complicated your facility is, the longer it takes you to turn around your facility and operate it. And so the beauty of the open ballroom on the right is being evaluated by more and more folks and considered by more and more folks. So it's a lot easier to get materials in and out of those facilities, but that would mean you would have to have a higher level of closed processing, because if you open up the spaces to each other, then you have to be concerned about virus clearance and reduction. So as our systems in bioprocessing become more and more closed, the possibility of ballrooms continues to improve, and the efficiency and smallness and logistical beauty of the ballroom will be further considered. Well, sorry for the formatting on the right. So what I've done here in this chart is to list these innovations a few examples of which I just went through. But there are others in process development, including high throughput screening, quality by design, hotspot landing pad expression systems, manufacturing ready cell lines. And in manufacturing, we talked about a number of these. In fact, I didn't mention reducing raw material variability as another pursuit to improve efficiency and reduce failures. And multi-product manufacturing has a very big impact on cost and overheads in terms of the operational efficiency of your facility. So innovations that lead to the ability to do multi-product manufacturing are very powerful on cost of goods. And lastly, the facilities, single-use modularity in facilities can help speed assembly, and closed processing in ballrooms, again, a very enticing, very high efficiency operation. The reason there are question marks on the right is there's no one answer, right? It comes back to your manufacturing strategy. What should you choose? It should be based on your strategy. So you have all these choices, and in many cases you will achieve the trifecta. In some cases you won't. But the pursuit of the trifecta is a good thing to go after. One example of a trifecta case, in which I like to call synergized manufacturing, is to combine single use with closed or connected processing, in some cases continuous processing, and put it in a ballroom. And if we can truly achieve a closed processing capability in the near future, this will enable multi-product simultaneous manufacturing. And if you can do simultaneous multi-product manufacturing, imagine the impact on your cost of goods per product. Very substantial. Okay, so to conclude, how am I doing on time, boss? Okay. So higher titers and single use, not enough. People are going beyond that. The manufacturing strategy is going to determine which tools you should use. The pace of innovation we're seeing is dramatically increasing, especially in the last 5 years, where all three things are being attacked, looking for those synergies. Regulatory agencies are generally supportive around the objective of quality. They don't care about cost, they don't care about speed. So target quality when you're talking to your regulator. And many of these innovations are still in development, very exciting time, and we'll see how they perform as these drugs and processes graduate to the commercial environment. Thank you very much.
Thank you, Parrish. Great overview of the technologies that are available to us to bring in innovation into our processes. And now, next speaker is Lynn Frick from Paul Biosciences. Many of you may remember Lynn at Tarpon Biosystems, which was recently acquired by Paul, focusing on bio-SMB technology, and before that, Perceptive Biosystems. But most of you probably don't know that Lynn actually got her start in the lab at Repligen, where we share a history of having contributed to the development of recombinant protein A, which at one time was a truly innovative new technology. Now it's a platform, of course. And without further ado, I'll introduce Lynn to talk about innovations in continuous manufacturing.
Well, now that I feel old, whenever you've worked at a product that is now off patent, that's when you know you've been around a while. So this is going to build on Parrish's talk, who's always a tough act to follow, in looking at some of the economics of what I'm calling integrated continuous bioprocessing. So I go beyond the words, perhaps, of process intensification, to really trying to envision in 5 years, this is going to happen, it's not going to happen overnight, but incrementally, how do we get to a fully integrated continuous processing with feedback to the feed stream, which is the holy grail of process efficiency in many other industries. And you'll see in the talk after this some of the information that Mark Brower from Merck has been working on and how you can actually use process analytical technology to help monitor these continuous processes. So we've already heard a little bit about this. This talk will focus a little bit on the investment. What is the payback? It's great to go and innovate and try new technology, but unless there's a true payback and benefit, the cost invested is never worth it. So how do we create a quality system, everything that allows you to still strive for this innovation. So I feel that one of the keys is building on what Parrish has in single use, and single use actually paired with continuous processing, they actually work hand in hand. As you create continuous processing, things shrink in your process. You use less water, less facilities, less tankage, less support. And because of that, you're then enabling an area of manufacturing that can, by definition, be more single use. So in a way, they're kind of linked together closely. Single use and continuous can create a process that truly does reduce cost. So we're going to talk a little bit, in this talk we'll focus on the economics of that. So first of all, a lot of people ask, what do you mean by continuous? And I think in this industry, which is both fed batch and perfusion, and there's a lot of different things you can actually have. This is a slide that came from the MIT conference on continuous processing. You can actually have example 1 where you have a perfusion, a continuous upstream, and a traditional batch downstream. You can have, as in example 2, where you're actually just using the capture step or the first clarification through capture as a continuous area. You can actually look at the entire downstream as continuous, or you can look at the whole thing as continuous. But in all these situations of process intensification, you always have to reduce costs in downstream, and the chromatographic capture step is one of the most onerous steps in terms of cost, whether it's clinical media costs, having to buy 60 liters of protein A and you cycle it 5 times, and then you're done with your run because you've made enough for your clinical campaign. So it's one step where there's the most inefficiency in the clinical process. How do you look at that and reduce cost in the clinic and try to shrink the process? So this involves reducing the total volume of the process, the chromatographic media, and eliminating tanks. In a lot of cases in a facility is a big cost driver. So just two technologies to introduce you to if you haven't heard of them, because it'll make the economic slide make sense. One is the cadence inline concentrator. So for those of you familiar with TFF, there's a single pass TFF technology, and this is now a kind of a pointer. The picture's up in the top. It's just its own unit, right? And it's got a valve on the top. It connects right in line, and really one way to think of it is just removing water from the process. Right? Anytime you remove water from the process, you're shrinking the whole thing. That helps the economics further downstream. So you get this compounding approach. When you remove water pretty early in the process, you've shrunk it enough that each subsequent step is a bit smaller, and that kind of cascades. And I'll show you an example of that in a minute. The second is what Tom just mentioned. This is a technology that came out of a startup, and I was involved with this from day one. The idea was there's a technology out there for chromatography called simulated moving bed. In a lot of other industries, petroleum, food and beverage, any industry where you have to make things at dollars per gram, they use this technology to help in the purification. Petroleum. So we were thinking one day about how would we implement this in biopharmaceutical, and of course the challenge in a manufacturing environment where things need to be sanitary is the valving, because in order to distribute a process across several columns or in a multi-column configuration, you need to use a lot of valves per column or per membrane or whatever you're running. So if you can make a disposable valve cassette in our case, or valve block, in this case it's the orange area you see on the picture made out of acrylic, then you have 240 valves on this one black, and that is a disposable. Anything that touches the product becomes part of the single-use flow path. So this is a fairly innovative idea, and this really opens up the world to single use in scaling down this chromatographic step. So we use the BioSolve model. This is from Biopharm Services that actually takes you through an economic analysis. It's a user configurable cost of goods analysis, and it allows you to put in your own inputs and look at different processes and see where the economic hits are. So just to give you an example, we looked at this process intensification with this inline concentration step, and these were the parameters we used for process input. It was what we call a generic MAB platform, so a protein A capture step followed by the subsequent downstream step. And in this case, we're looking to concentrate it 4 times. If you look at the typical flow path, and I won't go through this, but if you actually put the inline concentrator right in the middle of, at the end of primary clarification and prior to the downstream, you actually have an effect in the economics that's quite dramatic in terms of percent change in cost of goods. So depth filtration, sterile filtration, they drop by almost 50%, and then that savings is carried down the line. And this is all done through this analysis with the Excel program. So you can also think of this as a way to concentrate things in regular batch chromatography also. If you remove the volume, you're still having a dramatic effect on the size of the column or the mass that you have to process. So you're, again, shrinking the process by adding the inline concentration prior to the batch column. And you see the savings and resin cost in the bottom line. So to move on, how can you take some of this information in addition to inline concentration and actually couple it with some other steps to reduce cost in the most costly step? And in this case, ILC coupled with BioSMB multi-column chromatography can also improve the process economics even further. We've also looked at things, and some new technologies coming out of Paul soon will be continuous viral inactivation, continuous diafiltration, and other steps that will be incrementally introduced in order to, again, keep shrinking this process. So if you look at this picture, if the top line is a batch process where you have a 1-column process, you're actually running it, and one of the measures we use often is specific productivity, grams produced per liter of chromatographic resin per unit time, because it's a way to normalize the data. So with BioSMB, if you went to a 3-column process, you're already increasing your specific productivity at least twice, a little more than that, to 8 grams per liter per hour. But if you put this single pass TFF prior to the multi-column chromatography, you run a 4-column system, and then you're up to a much higher specific productivity at the far right. So if you're trying to get the most grams purified per liter of resin in a unit time, this is an option. And Mark will get into a specific example of this, I think, in his talk. Oh, there's my picture of the inline concentrator. So just to look at this, there's, in a very low titer process, this also can be applicable. Not every process that we deal with is a high titer process that's already moved on to the next generation. There's a lot of processes out there in perfusion that are still low concentration. And one thing we noticed when we started looking at this is this trend with savings over the batch. You can actually get this additional cost savings when you concentrate a low titer or a perfusion process up front. So these are just some of the ideas. I can go into much more detail, but I just wanted to lay out some of the economics of continuous processing to give you an idea. And we have an analysis tool if anybody has a process where they're running a batch process and you want to take a look at how either bio-SMB or inline concentration can impact that process. You can contact me and we can put you in touch with the right people to do that analysis. Okay, thank you.
Thanks, Lynn. All right, great. Thank you very much, Lynn. Well, so we've heard a lot about new technologies. We've heard about new strategies. So these are all critical aspects of innovation along with resources, but you also need something else. And what the other part of the secret sauce is you need somebody who's willing to be an early adopter, somebody who's an innovator. And Mark Brower is a great example of someone who's been willing to devote a significant part of his career to developing new process technologies. This takes a lot of risk because you really don't know whether these processes are going to see the light of day. And Mark's done a great job with implementing new technologies at Merck as a senior process chemical engineer. He joined Merck after getting his PhD at Cambridge University. Please welcome Mark.
So I think we have a really good introduction to the topic here. Today I'm going to focus in on the real-time release aspect of drug substance production and how the role of process analytical technologies and evolving process analytical technologies is involved with achieving that goal, because you can shrink a facility only so far by using these new concepts, and then at some point that's it. That's all you can get. And if you drill down deeper into the overhead and the operating costs of running a facility, it comes down to manpower and how can you shrink that cost in your facility. And when you look at that, there's a significant, more than 50% of your staff in a facility is QC. So if you can use analytical technologies in a smart way, you can achieve this overall reduction of costs. So, we've done a lot of economic analysis over the last few years at Merck, starting with a platform therapeutic protein process in a very conventional way. Very large 15,000-liter stainless steel vessels, stainless steel batch downstream. And, what we've shown is that those aren't economically viable, as Parrish has pointed out in his talk. So we started looking at the next generation of future concepts, and we started just by very, very, hybrid processes that were single-use stainless steel, and using again the single-use vessels and cell retention devices to drive up titer, and yes, you get a very substantial reduction in cost of goods. And a reduction of total cost of ownership of that facility. But when we drove a little bit deeper, we looked at these continuous type processes and seeing where can we go from just that batch mentality. And this continuous, you do get another jump shift in cost of goods and reduction of total cost of ownership. But again, to really realize that, you need to look at this real-time release aspect of the drug product that's being continuously produced by this process. And, we hope to even transform this platform in the future to a continuous process that can evolve to, newer type unit operations, some flow-through membrane perhaps that may be introduced that can further reduce cost of goods. So to demonstrate this process, a bunch of different companies have done this at this point with different feedstocks. What I'm showing here is an example of a fed batch bioreactor feeding a continuous process. We have all the different steps, centrifugation, depth filtration, column chromatographies, and single pass tangential flow filtration. And when you run these continuously, you can get yields that are very similar to a batch process, very high yields, above 75%. You get impurity clearance that you would expect, and very similar quality to the fed batch process when you're looking at it very specifically. But the interesting part is the part in the middle where we're showing how long each unit operation is being run in this process. So you can see each unit operation is started up when it's needed and shut down when it's needed. So you have a startup period in the beginning and a shutdown period at the end. But in the middle, you get a very nice periodic steady state. And what we've been focusing on is extending that steady state to multiple day, multiple week, and multiple month campaigns. And when you're running this mentality, you don't want to stop. You don't want to take a sample, send that out to a QC lab. You want to continue moving on. And the way to do this is process analytical technologies. So the vision of PAT at Merck is to engineer PAT into our manufacturing processes that will really give Merck a competitive advantage moving forward. And the mission is very focused on the manufacturing side, is to provide value to Merck manufacturing division with efficient commercialization and minimize inline manufacturing costs. That's what it boils down to. But in the middle, there's the development space, right? So the vision is very, very focused on this production aspect, but we have to tie it back into the development space. So some of the tools that have been implemented at Merck already are in online HPLC methods, spectroscopy methods such as FTIR, Raman and NIR, particle size distribution analysis, and that's on top of normal sensors for things like pH, temperature, and conductivity. And these have been applied to APIs at Merck. So one of our products, Januvia, has never seen a QC lab. It's just been released off of NIR. We're applying to vaccines for inline products to retrofit those, and are being applied to new biologic molecules on a daily basis. So what does it mean? I mean, we have to measure the process parameters and the CQAs of the product in a timely manner. And, from there, we go to two different ways. We go to the modeling aspect, which is understanding what the process parameters mean to the product quality, and then tying that into a model that would help us to optimize the process in the development space. And then from there, we go into the control aspect, where, the ultimate goal is to apply an appropriate control strategy to maintain a state of control in the manufacturing environment. So here you can see that the development use of PAT may be a little bit different than that in the manufacturing space. So to tie this back into the continuous processing, I'm showing this continuous process that I alluded to in the beginning, but have included here surge bags where we collect a very small bit of product from the previous unit operation before feeding it into the next. These surge bags become a place where you can implement PAT and to maintain, to check your process consistency and product quality throughout your continuous campaign. So things you may want to test for are, product concentration, the purity, impurity levels, the microbial load in that sample, and that's all necessary to run your process and to have a very high quality and consistent quality coming out the back. But then you need to address the release aspect, and those are compendial assays such as appearance and color, identity assays, and potency assays. And I think the mix of these two things, the parameters you need to control a process and the release criteria, can give you real-time release of the product. And while we're doing these exercises, we always ask, does PAT for continuous processing streamline real-time release? And I think we've convinced ourselves that it does. We have had to, because of the cost aspect of implementing a large degree of PAT, ask ourselves which assays are nice to haves and which are must to haves. and which assays can we combine to be multi-attribute methods. And I think that's going to be key going forward, is you can't have one method that gives you one output anymore. You have to have one method that'll give you several outputs to keep the costs down. And then you have the quality and regulatory expectations, which always must be met. So looking at the processes from the upstream and downstream side, on the upstream we want to monitor things, metabolites, the health of the culture, the titer. On the downstream side, you have a much larger list of things you may want to monitor, such as concentration, purity levels, quality. I mean, quality is a very broad category, but you have to cover the things you need to cover to get real-time release. So some of these technologies are ready to go. I think we classify online UPLC, HPLC chromatographic methods as something we can implement at the moment, but things maybe like NIR are on a 2 or 3 year horizon. Highlighted in red is the real-time bioburden readout, which we think is lagging even further behind, but is a critical piece to our real-time release strategy. So, in time, when all of these things come together, they can feed into your control strategy. So multivariate data analysis, adaptive control, and again, ultimately to this real-time release. So we've looked at one part of this triangle, the process monitoring, and that's one aspect of it. But there's also the critical inputs, and that's the raw materials where we see variability. So that also leads into the real-time release, and we need to develop PAT for quick release of those. And I think the two of those feed into a nice adaptive control strategy, which, if properly implemented, can get us to this real-time release goal. And again, at the moment, we're at this end product testing, and we want to transition that over a 3 to 5 year time frame to real-time release. And so now I just have one example. I was told to take another one out for time consideration, but I do think that the multi-attribute method, which I'm not going to talk about here, is important going forward. But now I'm just going to focus on this UPLC example. A UPLC is a chromatographic machine that has a pump like you're used to seeing, a sample manager which can sample from multiple different points in a process. The sample manager can also do automatic dilutions so that you don't have to be there as an operator to tell it to sample. And it can also have many different detectors connected to it, so you can use it in a multi-attribute way. I'm showing here some data that shows the dilution capability, where we get very good linearity of dilution from 1 to 30x. So you can sample at different parts of a process that may have different concentrations and still get an accurate readout of the titer. So what we did with this UPLC is connect it into our continuous process. We ran this for 30 days. We connected it into the bioreactor itself. We connected it to a permeate from a perfusion from a membrane. We connected it past continuous chromatography step, and again past a continuous viral inactivation step. And all of these were data was being collected 4 times a day for 30 days. And here's a select piece of that data. The top is showing the bioreactor piece, where initially we get very good agreement between online and offline capabilities, but we see some fouling of the probe. So this probe fouling issue is someplace we're investing so that we can eliminate this problem. On the downstream side, on the bottom graph, that's showing also very good agreement between online and offline measurements. So we see this online UPLC as a valuable tool for continuous processing and PAT. So, again, I think if we look back at the Trifecta, yes, I think we're spending money at the moment to invest in process analytical technologies. We're going to have to invest on the development side, but when it's implemented on the manufacturing side, there's a large amount of reduction in total cost of ownership and cost of goods that can be realized by the real-time release aspect. I think that's all I have.
Great. Well, thank you, Mark. Well, so now I'd like to open up the panel for questions from the audience. So please, if anybody has questions, I think the microphone there is working. Please feel free to step forward. And if not, I'm prepared with a list of plenty of questions to keep the discussion going, but it'd be great to have some more contributions if any of you feel so moved. Well, we'll just start actually with maybe a question for particularly Joanne and Mark. We've talked about the obstacles to implementing new technologies. It'd be great to hear about the current strategies for implementing new technologies within a biopharmaceutical company, and maybe also then what vendors can do to assist with that process as we start to bring some of these technologies we've heard about into the plant floor.
Yes. So this is a battle I fight every day. Right. So I mean, when we're talking about disruptive technologies, I think it's a lot to ask a large organization to jump cannonball into that pool. With our continuous strategy at Merck, we have a clear goal that we want to get to, but we also have a clear long-term plan that involves many steps to get there. And it's not going to be, we're going to build a plan and it's going to be continuous. And I mean, you really have to bite off manageable pieces. And so that may mean starting off in a continuous step or a semi-continuous manner and transitioning that forward. And, I think you just can't expect too much all at once, especially in this industry. Yeah. And,
Okay.
Make sense?
I just want to make a comment about our collaborations. I mean, I think that when you said about vendors, I tend not to even call them vendors at this point, because by the time you're dealing with a product that's finished, it's over. I mean, they help us. We work very closely with Millipore, Paul, GE, whatever, to actually help us develop the technology. They develop the technology, and we just feed in with information of, okay, this works, that doesn't work. And so these collaborations with the groups that develop the technologies are very, very important, because for disruptive technology, there's nothing really off the shelf that works up front.
Oh, please, Susan.
Very good talks. Thank you very much. So timing. So how do you assess what technologies you want to integrate into your core platform, and then how long does it take for you to actually complete that evaluation and implement?
So I think that feeds into the maturity of the technology, right? So I mean, if it's an innovative product, but it's GMP ready and you're ready to go, we can generate data pretty quickly and get that into a project. But I mean, if you're working with something that's a little farther behind, you may be fine-tuning a product and with a vendor, and then it may take a year or two for that to reach maturity and then to get GMP qualified. And so it's a balance of when you can implement that, right?
Yeah, I think, I don't know, I feel like we make it very, very difficult. I gave the example of our uber flexible facility. It was supposed to be very flexible, but it's actually, as a commercial facility, it's not flexible anymore. Getting technologies in and out of that facility is extremely cumbersome, so all the flexibility is gone. And I really wonder if there's something we can do ourselves. It's our responsibility to make things more flexible. Why is it so difficult to bring in new technologies? It takes an inordinately long time. The data that's required from both the group that develops the technology and from the actual the utilizer of the technology is huge. One of the things that you mentioned about raw materials, for example, if let's say it's a new resin or a new membrane, I think sharing the data from the manufacturer of the resin with the purchaser and the utilizer of the resin is going to be very important. I mean, for us, for vendors to share their databases with information for different raw materials and resins, filters, and things like that is going to be important. I don't know if this answers your question. But the more information we have, the easier it is to introduce a new technology.
Yeah. I think that's a good point. I think information sharing in a number of areas can really benefit us all, like leachables and extractables is a good example. And yeah, I think the time to implement new technologies is kind of a given in our industry. Maybe a question for Parrish and Lynn and is how does that long time gestation period impact the business of developing new technologies and bringing them to the market? How does it impact your outlook and approach?
I was going to say, I think both Parrish and I have the experience of being in a startup company because big companies often aren't innovative. They buy innovation. So we both, through Accelerex and through Tarpon Biosystems, were working on technologies that were truly different and innovative. And then you get to a point where you walk into Merck and you say, well, I'm Tarpon Biosystems and I'm a company of 10 and I'm going to sell something for manufacturing to them. You know, there comes a point where the business relationship can't work, that you have to be part of a larger organization. Now, Accelerex got much bigger than we did before we were acquired, but there is a point at which a new technology, you either have to create a whole new integrated bioprocess supply company, which is a very hard thing to do, or you have to be acquired by somebody who has the expertise in the worldwide distribution, and everything now is global. I mean, much more so than even 10 years ago, I'd say. You know, so nobody's just manufacturing in one country. You really need the supply chain across all countries. So I think for innovation to happen well, we have to continue to have big companies fund small companies and to have big companies that are our customers ultimately invest through new and enabling technology groups. This is the way Merck is set up, in new technology so that the small companies have a chance to be acquired by the big company. That's my solution anyway. It's all about the money. Follow the money.
Works for me, and after 15 years doing it, she's got a lot more hair left than I do. Okay. I think the cycle time in biotech is on the order of the cycle time for drugs. You know, because you develop the technology in laboratory, and then to really prove it out, you want to produce drug. That drug has to be demonstrated to be safe and efficacious in the clinic. And in parallel, if you're producing that drug with that technology, you're indirectly validating that technology as you're moving through the clinic. And ultimately, if the drug gets to market, and that's an if, right? It's 10%. 1 in 10 drugs get to market. Your technology will get to market. So, you know, it's a 7-year average, 7 to 8 year cycle. I mean, just look at single-use technology. It came out in the 90s, and the first licensure was at Shire in 2012. So that was, 15 years. So, I think it depends on the extent of innovation. If you take Mark Brower's case, where he's connecting everything, so he's going for the whole enchilada. It's a fantastic vision. But there are, just as he said very well, there are so many steps and such a long process. It's easy to implement in the lab and quickly, but when it comes to commercial, is it the right thing to do? Is it the right innovation to go commercial with? It comes back to your manufacturing strategy. But the cycle time is on the order of drug cycle time.
Yeah. Okay, great. Well, I think we have time for one more question. So we're going to challenge the audience here to be the last voice. Okay, if not, I'll take a stab at it. I think we've talked about the long time frames. If we think back 10 years ago, really there were no CSTR single-use bioreactors at any stage of implementation, and now look how far we've come. I think our industry can adopt new technology. If we think back 20 years, I think there were only 2 approved monoclonal antibodies, and now look where we are. If you think forward 10 years, what innovations do you think are really going to transform our industry in the next 10 years?
That was to me?
That's to all of you.
Yeah. I think, well, if you step back and look what's going on, the facilities are getting smaller and smaller and smaller, right? Okay, so let's extrapolate. Let's keep extrapolating. We've gone from 1 mega plant that serves the whole world. We're now going to distributed plants that are serving local markets. We're moving into cell therapies, which are much more local to the clinic, so we call that at-clinic, for-clinic manufacturing, even smaller type operations. Cell therapies are made in 1 to 10 liter unit, size batches. And as drugs get more potent, at some point we'll be manufacturing drugs at the micro scale, and it's dosed, through your iWatch. Extrapolate, I think in 10 years we could see that. Those technologies all exist today. Microfluidics, chemometrics or combinatorial chemistry metrics and microfluidics combined with analytics. We could see, micro manufacturing and dosing on person, for person manufacturing.
There you go.
Mark? Joanne?
Well, I was going to go to the custom, the affinity ligand type route. I think, as we move farther away from our customary antibodies, that we need those, new modalities to really help us with high throughput processes.
Joanne, Lynn?
Well, I kind of agree with Parrish. I think that the future is something that we're going to go to more custom drugs made for specific indications, small amounts, but we have to be very flexible in terms of what needs to be made and how it's going to be made. So I don't know if the future is more monoclonal antibodies.
No, I agree that it's more personalized medicine. I was talking to a stem cell guy this morning, and I think that we've already been asked as a continuous processing supplier to get smaller with the equipment in GMP. So the question I just got last week from a company that's been working with continuous for a couple years, what is the smallest GMP system that you're envisioning in the next 5 years? Because we want it small, because it's going to be very specific to a particular process. So I think shrinking processes and getting more into personalized medicine, novel manufacturing.
DARPA is funding a program right now, have been for a couple of years, to make a laptop-sized drug manufacturing device for the medic in the field. Currently funded.
All right. Well, with that, I'd like to thank you all, and let's thank our panelists one more time, and thanks for your attention.